in order for us to stay funded, we have a couple funding agencies. The two big ones that have been behind us the whole way, and we're super happy about that and dependable, have been a program project called IDAT, which is a sort of technologies grant run by BER. And if you guys have any results that are relevant to environment, climate change, synthetic biology, engineering, this grant really wants to hear about that. And the more of those kinds of works that we do, the happier they are. We're also funded as a core for DNA repair projects. So you'll notice that Mikkel's like a super expert in DNA related stuff. And that's partly because of this DNA repair project. And again, just want to mention that John got all these, funded to begin with. So, he had the vision to put this together. We also have a user facilities grant. So from NIH, and again, this is a number crunching thing. This is like, how many papers have you published? How many users have you served? So that's why all the citations to us are really important to continue these fundings. And, if we go down 1 year, they ask questions. We also are very happy to receive some short-term industrial projects. Sometimes, they come through, and then there's also some small short-term grants, like little LDRDs and things that we get. Not dependable, sometimes 6 months, just a little bit of cash, but we're always trying to support the beamline in some ways. And I think the science that you're going to see and we've produced, is sort of I think it's a lot of exciting stuff going on. And we are looking for postdoctoral fellows, and hopefully you see and recognize from the work that Mikkel and I and others have been involved in, that you'll see over the day. It's not just a beamline job here. We're not talking about just helping users out. All of us have really exciting and interesting independent projects, and there's room for that. So if you guys have somebody who's looking for a postdoctoral fellow, or you yourself is looking for a postdoc, please respond to a call at LBNL hiring for the beamline. All right, so on to the exciting stuff. This beginning part here is kind of an intro to SAXS and the ALS, and I'm, cognizant of the fact that some of you have never been here or been to a synchrotron, so I'm just gonna do some intro stuff here. So the advanced light source here is pictured in the middle. It overlooks the Bay Area. It, I think, was designed in copy to the geometries of the Parthenon in Rome, and that was actually, funnily enough, a kind of a light experiment where, depending on how the light came in to the hole in the top, that helped them decide when to plant, what season it was, and in different places along there's altars to the different gods of the seasons and stuff. So I like to think of the ALS a bit like that. Instead of shrines to different gods, we have sort of shrines to different imaging technologies that look at different aspects of matter and physics and biology. The one that we're going to talk a lot about today is the small angle X-ray scattering, and I'll show you updated pictures of that. But we also have very close partnerships with X-ray tomography, which can look at whole cells, and also at crystallography. So this is sort of the atomic level nature. This is sort of solution and macromolecular complexes, and then this is how the macromolecular complexes are acting in the cells. So sort of a whole scale. And if you want to bridge any of these spatial scales, please come talk to us. We love that kind of science as well. All right, so the small angle X-ray scattering beamline. We've updated it quite a bit. We now have really focused in on size exclusion coupled SAXS, which is this instrument here, which Mikkel and Daniel will be talking to you a lot about. You can see the columns here in the sort of chilled area here, and then, you know, a loader and pumps and stuff. So those all feed sample into the X-ray beam, which you can see the position by this bright light that's shining on the sample. Zooming in a little bit closer to that deck region, so that little light there is this guy right here. You can see more details about the robot deck. You can see the robot needles, which I'll talk about later today. And then here's the sample position with the camera that's jumping up, hitting this mirror, and then looking at the sample. This is one of the liquid handling robot needles. The SEC stage is on the side here. And this can translate between the two modes. Daniel implemented this sort of translation a little while ago. So we can, we've been running SEC high throughput every week, and this sort of transition requires about an hour or so, depending on how challenging it is that day. So, and those are the things that we're really been just working really hard on without looking at too much of the time resolved or other technologies, though we're branching into those too. Besides, you know, those brands, besides serving, you know, the people, the hardware here, we also have a bunch of web services that I hope you'll be aware of, and some of which we're going to talk about a lot today. So if you go to our SIBYLS.als.gov, you click the SAXS button, you'll find these web resources here for helping you analyze your data. So that's basically what we're hoping to continue to fund, and can only do that with the science you guys do. Oh, the one last thing that I would like to mention is that we also provide these workshops. So this is the 12th annual workshop and at the ALS user meeting, but we also have another one in the middle of the year related to SPDR. This is the second one that's been over Zoom. The last one we did last year was actually pretty good. I hope this one can meet that. It felt good anyway. And it's obviously hard to tell without being in person. That's the trick, right? With all these Zoom things.